All right, before we move on to the second major step of respiration, the Krebs cycle, I just wanted to make one more little note. When we're moving into the mitochondria, sometimes this is called the transition step. So if you ever hear that term, transition step, it's just this. All right, Krebs cycle. First thing I want you to realize about the Krebs cycle, it's also called citric acid cycle. Not a big deal, but you may hear them used interchangeably. This is happening inside the mitochondrial matrix. So now we're not only inside the mitochondria, we're at the very, very center of it, inside both of the membranes. Now, there's a molecule in there that is just there, okay? Just accept that it's something that is there and present all the time and is being used for this process. It's called oxaloacetate, and it's a C4 molecule. There's a bunch of other hydrogens and oxygens and all that, but it's got 4 carbon atoms. Now, what's going to happen is we're going to take our acetyl-CoA that we just made in the transition step. Now that we're inside the mitochondria, we can actually drop the CoA. We don't need it anymore. And so we're left with C2-acetyl. So just a reminder, our acetyl molecule has 2 carbon atoms. We're actually going to take that C2-acetyl and we're going to combine it with the C4 oxaloacetate. Do a little math here. 4 carbons plus 2 carbons. That means we're going to end up with a molecule that has 6 carbons. It's called citric acid, and that's why sometimes it's called the citric acid cycle. Now, ladies and gentlemen, citric acid is the stable molecule of this process, which is why it's named that. So we're going to run C6 citric acid through this Krebs cycle, again, with the whole goal of trying to pull out some energy. The first step is we're going to go ahead and pull out 2 carbon dioxide molecules. If you're taking away 2 carbons from a C6 molecule, that means we're going to be left with a C4 molecule. Don't worry about names. We're not really concerned with that. We're just going to keep track of the carbons. At this point, we're going to be able to create a little bit of energy. Not a lot, but we'll be able to take 1 ADP, turn it into 1 ATP. Next thing we do, we're going to remove an electron. So don't forget what kind of reaction is going to be exchanging electrons back and forth? Redox. Who is being reduced in this situation? In other words, who's getting the electron? It's going to be a molecule called FAD+. FAD+ is like a cousin to NAD+. Don't worry about differences, just accept that it is another temporary energy storage molecule, and by giving it an electron, it turns FAD+ into FADH. So who's being oxidized? Our C molecule right here. Remember, that's what we're tracing through this process. So we have a little redox reaction. Now we're going to do another one up here, except this time we're going to remove 3 electrons. So another redox. Our C4 is the molecule being oxidized. And now, back to our old friend, NAD+. NAD+ will be reduced. And we're going to turn 3 NAD+ into 3 NADH. Now, even though there's actually a lot more steps to this process, a lot of different names, these are the major ones. Time we've run through this entire thing, we have regenerated our C4 oxaloacetate. This can go ahead and do the process over and over and over because we keep regenerating it. It's never used up. So a quick recap. We take our acetyl-CoA, drop off the CoA, and now we're left with acetyl. We combine acetyl, which is a C2 molecule, with oxaloacetate, a C4 molecule. That gives us C6, citric acid. We're going to yank out a couple of CO2s, which leaves us with a different C4 molecule. Create a little bit of ATP, do two different redox reactions where we yank out electrons from our C4, which means it's oxidized, and we're going to give them to FAD plus and NAD plus, which means they're going to be reduced, and that gives us some FAH and NADH, storage molecules for that energy. We've regenerated our C4, and now all we need to do is do it again. So let's take a look at our output box. CO2, NADH, ATP, FADH. All four of those things we're going to be creating. But you're going to notice I only have two CO2s here. How come I've got four up in the box? Don't forget, we came into this situation with acetyl CoAs. We just showed the process for one of them. So guys, everything you see here is going to occur twice. One time for each of the acetyl molecules that we made in the last step.